So this session we have four papers. The first paper is from Professor Chattopadhyay sir from BCKB West Bengal. Second one is from Dr. Upendra Kumar sir. Third is from Madam M. Sahu. Another one paper is from Dr. B.K. Vindhani. So may I request Professor Jogi Naidu sir, please chair the session. Professor Naidu sir from Naira College, Professor Agronomy. He will chair the session. Next, may I request Upendra Kumar sir, please. Then may I request Chiranjeev Mandal, please make the session as a rapporteur.
Hello, good morning to all of you. Respected Vice Chancellor of this university, Mr. D.N. Rao Garu, the other dignitaries, the scientists from across the country, and of course our own colleagues and scientists from our Acharya N.G. Ranga University. I welcome all of you. As you all know already, the senior persons are already discussed and given some introduction about this sustainable agriculture, which is a very, very important topic nowadays. After the inauguration is completed, now just we will start the actual technical session. I request all the students, especially students, and of course the staff and other, you try to just concentrate on the topics which are being carried out by each and every scientist, which is very, very useful for you, especially the second year and third year students. I request all of the students maintain discipline and try to just focus on the what is the actual sustainable agriculture. As I said, many of people morning, is a wide, broad term which cannot define in a single word or a single sentence. So this is the opportunity for all of the students, especially the opportunity where many scientists are coming over today and they want to deliver their lectures. So let us start the technical session with the first one, Dr. B. Chattopadhyay, professor from BCKY. He will present the community seed production, a new vista for seed security. So this is one of the, as you all know, one of the basic input is seed. So we have to start from that input, that is a seed.
Good morning once again. My topics is community seed production. This is the new vistas for seed security. So all of you know that gradually our farmland is decreasing and we have to produce 470 million tons in 2050. So until and unless there is a seed security, how do you think that you will give more attention and food security? First slide, please. First. So quality seed is the basic input in agriculture and vehicle of enhancement of agriculture production. So if you put the quality seeds, you can easily enhance 15 to 20% productivity in your fields. Just you change the farmer safe seeds to quality seeds and keep the all agriculture practice farmers commonly used in the previous years. It will give you a 15% productivity enhancement. So quality seeds, you have to ensure the quality seeds at the doorstep of the farmers. So all of you know. But it's our irony. Next, please. Next slide, please. It's our irony that after the completion of National Seed Project 1, National Seed Project 2, National Seed Project 3, this National Seed Project is already completed. Still in India, farmers are not getting the quality seeds. Only 25 to 30% quality seeds farmers are getting. If the situation is like this, our national seed project under this, all of you know, the State Farm Corporation of India, National Seed Corporation, and all the state seed corporation, they were entrusted to supply the quality seeds to the farmer. But in 2015 also, our seed replacement rate of cereals is meager. That means still in India, whatever the cultivation is going on, the 70% seeds is from farmers to farmers. Only 30%. So if the saved seed or farmer saved seed is 70%, so why don't you entrust the farmer community to raise this quality seed 70% and 30% by government support, like your National Seed Corporation, State Seed Corporation, and SFCI. So the gap between this gap between demand and supply is the major constraint of productivity enhancement. So we are talking about the enhancement of productivity. We are talking about the sustainability of our productivity. I don't know what is sustainability. So 2000 kg per hectare yield is if our productivity, then can we agree with me that our sustainability of productivity will be 2000 kg per hectare, or it will be grow up to 3000 or 4000 kg per hectare like other developing country. So this sustainability, I don't believe this sustainability is a stable or rigid term. It is a flexible. Today population is like that. 10 years back, 10 years it will be double. Then sustainability of productivity is like a big word. Sustainability means must be changing. Then how this gap will be bridged up. Next slide, please. So the farmers of this your agriculture who are actually so this is actually seed scenario in West Bengal. Just I put this slide for a situation in Odisha. Odisha is like that. Only 25 to 30% cereal seed replacement rate is there. Our West Bengal is 54 lakh hectare area is under rice cultivation. If you put this 30 kg per hectare seed rate, then we need that 2,90,000 metric ton of seeds. But only 1 lakh hardly last 15 years, private sector, public sector, their effort, only 1,23,000 tons West Bengal is producing the rice seeds. Wheat, maize, all seed replacement rate is not at par our projected seed replacement rate. Next. This is actually seed replacement rate, SRR. Oh, sorry, this is the printing mistake. Seed SRR, previous one. So next, please. So what we will do? This is the problem. So then in everywhere, in every state, you have to identify its seed production zone. Where will it grow? So for my experience and knowledge, your Kalahandi is a very good area for especially rice, hybrid rice and vegetable seed productions. So any state you have to identify the seed production yet and create the infrastructural facility. And really I am happy I am working in this state from last 5 years. I am happy in Kalahandi, there are a lot of seed processing plant is already established. Some of the rice mill storage go down is already. Second is to ensure the supply of situation-specific crop variety, breeder and foundation seeds. This is the basic seeds required for quality seed multiplications. So these you have to ensure quality breeder and foundation seeds. Community seed production and establishment of seed village. All of you know the Indian, after 2003, seed village is a policy in our national seed project, seed policy. And now Bangladesh is also going to establish this community seed production or seed village. So seed organization of public and private sector should be take initiative for rapid multiplication. And what emphasis I have to given this involvement of large number of farmers cooperative society and NGO in seed production to explore the possibility of homestead courtyard seed production of vegetable seeds like amaranthus, spinach, okra, laplam bean, gourd and pumpkin. Odisha and Chhattisgarh is and West Bengal, this eastern part, mostly tribal populated area. So in tribal area, tribal village, these vegetable seeds you can easily produce with the homestead. Simply your basic set you put every family and allow to grow there and allow to use the plant material or fruits only like your gourd. You allow only 2 fruits per family. In 200 family, you will get the 400 fruits. You bring back to your company's plant and you extract your seeds like that amaranthus, spinach. You can easily multiply your seeds. Next. For good quality improved seeds, the emphasis should be given on this. Keeping on every state that you have to increase the seed replacement rate. That is your duty. Proper technology should be adopted the post-harvest handling of seeds. This is my practical experience in National Seed Village Policy, where the farmers are eager, farmers are producing the quality seeds. But after harvesting, the post-harvest handling, there is no support of government, there is no support of anybody. So that is the, again, the setback. Quality seed produced by farmer and farmers are selling the quality seeds as a food grain. So this should be taken into consideration, that post-harvest handling. Scientific storage go down. Nowadays, you know, the India government is giving the RIDF support, Rural Infrastructure Development Fund. So on that farm, farmers can utilize with this storage go down. And for productivity enhancement, this line should be kept in mind, the distribution of quality seeds in time at the doorstep of the farmer with a reasonable price. In our state, the government support what actually happening, the quality seed is distributed, but not in time. That's why the farmers are forced to sow their own seeds, though the government quality support seeds are available 7 days or 10 days after sowing of the crops. Technology should be adopted for enhancement of seed quality parameter for better emergence of high vigor seedlings in the farmer's field. So not only the quality seeds, you have to ensure the 6 quality parameter of seeds at the time of germination in the farmer's field. That will be the actually success of our a good high vigor seedling established in the high vigor seedlings. Next. Next. And now, how you will do this community seed production program? The model is every village there should be a apex body like NGO, KVK, Panchayat, Growers Cooperative, Growers Club, or Primary Agriculture Society. In every village in this eastern state, there are a lot of NGO. I saw in this Mala premises one NGO is working in Kharagpur and also working here also. Supporting institution, Agriculture University, Horticulture Department, KVK, even if the teacher of agriculture school, he is a technologist. So these are the supporting institute. Participatory grower cluster at village level. Can you project the last slide? Last. So this is the model how you can grow the community seed. This farmers participatory seed production, the apex body should be Samavai Krishi Unnayan Samiti, Farmers Club, Self-Help Group, Panchayat, NGO, KVK. This organization can play as an apex body and helping farmers is scientists, technologists from SAU, State Agriculture University, KVK, departmental staff, agriculture teacher of the school. These are both ways. The farmers should be selected by the apex body from different village, say village number 1, village number 2, village number 3, village number 4. And they will arrange the training because farmers know how to produce the crop. They don't know the seed production of crop. Only technology is given. The seed production technology, not all through crop production technology. So the farmers know how to grow rice. Only you have to teach the institution or duty to teach the how to produce quality seeds. And this must be in a cluster level. Go for the first one, the duties. So there are 3 components, 3 bodies. One is apex body, another is the farmer who produce the seeds, and another is the institutional support for the production of these quality seeds. So these 3, they have their duties to perform or successfully performing for establishment of community seed production. So apex body, if no the cooperative society, NGO, KVK and farmers are apex body, then identify the farmer, assess the crop area and assess the demand of the seeds of that particular village. Suppose a village needs 10 tons of seeds for Kharif seasons. So your target of production is 10 tons of seeds. So that has to be decided by the apex body. And second, identify and select the energetic, educated farmer and train them by as a master trainer of this. Technical guidance of Agriculture University and institutional. To link up the trained farmer of the Agriculture University institution for further query, motivated and link up the farmers of seed cluster production level and supply of foundation seeds and necessary inputs. This is a vital. It will be failed this project if you cannot supply the foundation seeds and necessary inputs. That means foundation seed is the basic seed for certificate seeds production. To arrange periodical field inspection by seed certification agency, this has to be taken by this apex body. otherwise seed will not certified. To arrange the scientific seed processing, storing, packaging facilities, and to arrange periodic checking, so storage go down of the seed expert. Next. This is the duties of KVK Agriculture University people, to train up the selected farmer on seed production technology, to ensure frequent visit of the expert to seed production plot, to make effective group discussion, seed grower, solve the current problem, and inspect the field association with the team leader, to monitoring rogging and post-harvest operation, fix up procurement price seeds, and consultation in consultation with appropriate authority. Next. This is the duties of the farmer, participatory farmer. They have their responsibility. The field cluster should not be less than 3 acres. Seedlings should be collected from the community seed bed. Why this is actually in the eastern part, the holding size is very small. So that's why if you want to ensure the high vigor seedlings, so make it a community seed bed for each cluster and allow the seedling to transport that individual farmer's plot from the community seed nursery bed or seed bed. Field clusters should have assured irrigation, isolation should be maintained, periodical rogging operations should be carefully followed. Care to be taken in the threshing floor, drying floor, avoid physical admixture. This is our practical experience. Whatever the technology you are given, but only lack of their care, they usually mix up the floor in their threshing floor by 2 or 3 seeds. Seeds should be dried optimum moisture level for submission of approximate. Next. Field cluster, what I have already mentioned. Next. So this is actually experience, what I have already told. So this is just like a theoretical. So one Gotra Samavai Krishi Unnayan Samiti, this is in the district of Nodia. So we started to establish this community seed village, seed production in 1997. So from 1997, just I will show you how the farming community can establish themselves as a seed entrepreneur. Next slide. So this is actually a figure that in 1997, only 22 farmers is associated with the seed production. In 2015, now we have 15,000 farmers are associated with this seed production program. Total seed production we started with the 6 hectares of land. Now it is 550 hectares of land under this cooperative society. Targeted seed production first year, we started with the view that that village required the 20 ton seed production. We achieved in 21 ton seeds. Now it is 300 in 2014, it is 3,000 metric ton quality seeds. Next. So why this seed production, community seed productions? Only to ensure the seed supply chain, only to bridge the gap of demand and supply. No. There must be other objective of this model. That means the employment generation through seed production. So in a thumb rule, if you produce 300 metric ton of wheat or rice seeds, so you will ensure 40 to 50 mandates per day. So that employment generation, so first year in 1997, we hardly able to generate 230 mandates and 112 women's mandates. Now in 2050, it is 20,000 mandates and women's mandate is like 4,000. So you just see the table, how miraculously you can increase the rural employment through seed production, whatever may be the seeds. And the capital generation, first year in 1997, the cooperative gained only 2 lakh rupees out of these 21 ton of seeds. Now, 3,000 metric ton, the total income generation, income is turnover. Turnover is 4.5 crores. So 4.5 crores is a turnover. So if you take the 10% of profit, what amount of profit the farmers can get if they aggregate, if they form cooperative and go for the seed process. Next. This is actually the social impact in village, because there is a hue and cry whether agriculture will be the prime focus or industry will be the prime focus. In our state, there is a really still today debate is going on, whether agriculture is an important thing or industry is an important thing. So in this level, they started with the 20 ton, and now in India, they produce 300 metric tons of foundation seeds, that to 30 variety of rice. So you cannot get 30 variety at a time in any seed organization's places. Hardly you get 4 or 5 variety. Next. So this is the actually learning experience of this, that this is the core area where just you can generate money and you can just give the assurance to the farming community that through agriculture, through seed production, you can enhance your socio-economic and even uplift your socio-economic upliftment. So with these few years, I think this is next. So with these slides, I thank you all of you for patience hearing. Now any question if you have.
And he has given a lot of information regarding seed. As students, as far as students are concerned, one has to know there are so many categories of the seed. As sir said, there is a breeder seed is there, and nucleus seed is there, and foundation seed is there, and certified seed is there. So seed is, we are government organizations producing only 10 to 10% of the seed. The remaining seed is only meeting from the farmer to farmer, village to village. So this is one of the very critical information sir has given, and students has to learn about the only seed importance, the quality of the seed. You see, some of the districts in Andhra Pradesh, the farmers are quarreling about the for want of a good quality seed. So that is the very basic input. One has to verify, get the good seed, since the cultivation is starting with the very basic external input seed. So thank you very much, sir. You have given a lot of information. Any questions are there, you can free to ask and free to interact.
Sir, I just want to know that I have one doubt. Really, it is a good thing you are informing the seed quality. But there are so many seeds. Let me know about anything, information with you about the recalcitrant seeds or orthodox seeds. Is there any options for promote the seeds?
No.
So many
This
seeds
is
having recalcitrant.
This is a character. This we when we teach to the student the classification of seeds. So depending upon the storage life of seeds, that is physiological basis, one is recalcitrant seed, other is orthodox seeds. Orthodox seeds means the seeds we can reduce the moisture level and we can store for a higher longer period. But recalcitrant seeds, you cannot reduce the moisture. So you have to maintain 25 to 30% moisture for recalcitrant seeds, like all the tropical forest trees, rubber, then cocoa, coffee, mango, jackfruit. So these are the recalcitrant seeds. Only you have to conserve with about 30% moisture. But that to know. But
Is
like
there
that,
any study you have done it? And is there any study so far for recalcitrant seeds or a dormant seeds?
recalcitrant seeds, there are a lot of methods, right from cryopreservation to transport for moss. Suppose you want to send rubber seed. Rubber seed is as only 15 to 17 days is your viability. Sal seeds, it is very less viability period. Even tropical rainforest, if you notice, the fruit drops occur when precipitation starts. That means when your rainy season start. In tree, it will remain 12 months, 14 months. After that, it fall down from the tree when the available nature fix such a condition that at that time the oxygen will be available, light will be available, and moisture will be available on that nature, then new offspring will come off. So recalcitrant seeds and orthodox seeds, there is no relation in seed trade. It is only the storage behavior of the seeds. It may be rice is your orthodox seeds, your rubber is a recalcitrant seeds. So if you do business in rubber seeds, so you have to cautious about this. How long, how far you have to transport, transit period, 17 days.
Okay, sir, thank you.
So now I request Sri Sahu sir, is from NIT REL, Sahu madam, sorry. So she is going to present the community forest rights and community forest management, a case study for sustainable development. So you may think how the forestry is related to sustainable agriculture. So definitely, forestry management is also related to sustainable agriculture. You know the effect of deforestation, you know the advantage of forestation. Now I think it is also a relevant topic with relevance to the sustainable agriculture. So let us see.
Well, it's afternoon now. Good afternoon, everybody. Respected chairperson, dignitaries, and students of Centurion University. Well, I take a great pleasure to be here today to present this topic. When the entire world is running after IT and different technical things, here we are talking about the real thing, the sustainable, agriculture and food security. Food security, which is one of the first agenda of Millennium Development Goals. Before I start with my topic, I would like to give a short introduction how I come across community forest right and community forest management. Well, this is the work I was doing with University of Toronto, Department of Forestry. During my work with Vasundhara, it's an organization, it's a research and policy organization based in Bhubaneswar. I was part of the research team working on, implementation of Forest Right Act in Orissa. And that's how I thought this would be the best topic to be, here and speak about how forestry is important for the food security. I'm from NIT Rourkela, Department of Humanities and Social Sciences. Let me clear, I'm not an agricultural scientist, since there are a lot of agricultural scientists and technical people here, so I'm basically an anthropologist. So my perspective will be more on community side than talking about the technical part, as Professor Upadhyaya gave a lot of technical things about his topic. The overview of my presentation would be the introduction and India and Forest Right Act. Since I'm talking about the community forest right and forest management, it's directly related with the Forest Right Act and how India came up with the Forest Right Act. Community forest management versus joint forest management. I'm definitely talking about a case study of Budi Kamari, how the Budi Kamari people managed to, manage the forestry and made their livelihood sustainable. Well, before I start, how important forest is in our life. Forests contribute a lot in everybody's life. We are not, some are indirectly, some are directly. We sitting in the urban, in an urban sector, we never really realized forests could ever, contribute to our life. But forests do play an important role. As I said, it is one of the big first agenda of Millennium Development Goal to eradicate extreme poverty and hunger and in striving for food security. Globally, millions of people depend on forests for their food. We all have a misconception that forest is only for the tribals and the marginal communities, but it's not. We all are actually depend on forests. Different countries have recently adopted diverse approaches to increase food security and thereby concerning more on agricultural solutions. Well, we are talking about the agriculture sustainability and food security. So forest has a bigger role to play for agriculture production, forest production, and promoting agroforestry systems. Well, how Forest Right Act came? India actually was dominated of two important laws, that is Indian Forest Act 1927 and Wildlife Protection Act 1972, when large number of forests was taken care by the government, and whereas sidelining the actual forest dwellers and forest communities. So how these people are going to get the right? It's been a long struggle for the community to get this right, to actually hold the forest on their own hand. The post-colonial Indian state reinforced, centralized, as I said, large number of forest was controlled by government, thereby having National Forest Policy of 1952, which focused more on promoting forest resources while commercially exploiting the Minor Forest Produce and Forest Conservation Act of 1980, thereby evicting large number of forest dwellers on different development projects, may it be the dam or may it be mining, may it be the sanctuary. So there were a lot of evictions happening in until 2000 when actually the large communities, the forest communities just struggled, and they started with with a revolution of having their rights on the forest. So the Forest Right actually, so as I said, they united with the social movements working with forest users across the country, mobilizing them to raise their voice against the demeanor of democratic right to life and livelihoods to the vast tribal population. The strident opposition led to the formulation of a new Scheduled Tribes and Other Traditional Forest Dwellers Act in the year 2006 and came into force in 2008. So what this Forest Right Act actually is about? Forest Right Act is giving an individual right on the forest for who are living in the forest or using the forest land for ages. And it also gives community forest right to those forest dwellers who are using and relying on the forest for ages and ages. The FRA, Forest Right Act, provides the resolution of traditional forest rights of forest dwellers across India, including individual rights to cultivate land in forested landscapes and community rights to control, manage, and use forests collectively. Forest Right Act recognizes and wastes secure community tenure. As I said, it also includes the community forest resources, which commonly defined as forest land within the traditional or customary boundaries of the village or seasonal use of landscape. In case of pastoral communities, including the reserve forests, protected forests, and protected areas such as sanctuaries, national park, to which the community had traditional access. So this Forest Right Act actually gives access to the forest dwellers who are also living in the national parks and, which is popularly known as the restricted areas. Well, what these community rights actually give? There are a number of sections which are giving rights to rights on their land and their customary lands and the resources. As you all can see, subsection 1 of section 3 of the Forest Right Act confers the right of ownership and access to collect, use, and dispose of minor forest products traditionally collected within or outside the village boundary. Again, it includes the MFE such as brushwood, stump, cane, tussar, cocoons, honey, wax, lac, tendu, or kendu leaves, medicinal plants and herbs. So they actually have legal rights on all these resources in the forest, which community rights are given under the Forest Right Act. There are other subsections as well that covers the primitive tribal groups. Yes, the primitive tribal groups also has important role to play in forest, where in one of the section of Community Forest Right Act, where they've been actually should be given the habitat rights in the forest. It also covers also the disputed lands under the nomenclature in the state where claims are to be disputed. Covers rights to convert pattas, lease or grants of forest land issued by local authority of state government into the titles. So basically, these are all the titles they should be given under the Community Forest Right Act. So this is the CFR status of Odisha. As you can see, government has is very clever at the end of the day. Even if they have the Forest Right Act, they are restricting people to give rights on the forest. As you can see, very less number of district has been given the community forest right. So the total is 12,500 claims were filed by the Grama Sabha in the villages. Well, let me also tell you one important part of Forest Right Act. It revisionates the rights of Grama Sabha of playing an important role, where the subdivisional level at the block level used to go through all the files, and then it again goes to the district level committee, where the rights actually looked into thoroughly, and the rights is finally provided by the district level committee under Forest Right Act. So out of which 12,500, 3,474 CFR rights were given, which makes 7,702 acres of forest lands. Well, this also includes the entire the total number of districts of Odisha. There are the possibilities of rejection as well. If the community is not giving the details of what are the areas they are accessing and all the documents they are not providing, so their rights can be rejected. So total 301 CFR rights were rejected as per the Ministry of Tribal Affairs 2014 document says. So now, community forest management and versus joint forest management. As I said, government has always been a big threat to community. So they also never leave the community as themselves. There has to be a government to be to be part of the forestry. So for that, in every district in every villages or 2-3 villages, there has to be a joint forestry, that is the Vanasuraksha Samiti in Odisha. So what this Vanasuraksha Samiti is actually doing to community forest management. So as I earlier told, there are large number of people who are depending on forests. Odisha in particular have thousands of villages are involved in active protection and management of forests. Most started protecting on their own initiative as a response to forest degradation problem. Odisha has been struggling with the process of formalizing these initiatives and bringing them to the joint forest management fold. Now, when this joint forest management came into force, how difficult it became for the community people to accept and feel that this does not put the management powers and decision making the hands of local communities. So tension between the formal JFM system and locally evolved. As I said, the large number of forest resources were taken by the Vanasuraksha Samiti than by the community. So that becomes a problem, and the problem of dichotomy between the informal community initiatives and JFM has been intensified due to the lack of sufficient efforts by the state and forest department to address this problem and take up trust building efforts. So, well, there is always a lack of leadership and proper guidance by the forest department and proper involvement and participatory approach by the forest government as well as the involving the community. Now coming back, how community forest management has actually played an important role in a place called Budi Khamari, and sidelining the joint forest management. They have taken the entire care of the forest by themselves. So let me start how actually it is one of the one of the oldest form of community forest management in Odisha. It started like this in 1952 to 1972. The reserve forest of Budi Khamari were managed under the Kopai Circle as per the forest department working plan. By the early 1980s, the local population started experiencing the scarcity of resources. Budi Khamari is placed in Mayurbhanj district, and it's between the Betnoti and Baripada. In 1983, the first hamlet level forest protection started. The first step was taken by banning on extraction from the forest. 5 to 7 people started patrolling the forest every day in the Thengapadi mode. Thengapadi is very oldest mode of protecting the forest. As you can see, two Thengapadi forest protectors are standing, and they look after the forest, and how it became a proper system in Budi Khamari. Initially started with 5 to 7, started with the forest, 5 to 7 people patrolling. Then with the passage of time, villagers were allowed to collect broken twigs and dead branches from the forest protection with the permission of the forest protection committee. The members traveled by bicycles and on foot to other villages surrounding the forest to pursue them to adopt the similar protection measures. Then rules and regulation were also formulated by the forest protection committee for benefit sharing as well. The collective effort of the villagers resulted in a visible gradual generation of the forest. So how the 5 to 6 patrolling of people with the time, there was a regeneration of the forest. In 1987, a meeting of leaders of 70 villages were organized for creating awareness about the need to protect the forest in Hatikot village. Hatikot is a nearby village. A dire need was felt to create a network of village community to address the issue. So with the time, there were a large number of villagers who joined this forest protection committee. So in 1988, formally the Budi Khamari Community Forest Protection Committee was formed with 20 village forest protection committee. So there were 20 villages who were part of this committee to strengthen the protection system collectively. A joint mobile patrolling force with 100%, where 5% from each village was constituted to patrol a large area. The forest department was not. As I said, the JFM was one of the 3-2 community here. The forest department was not involved in patrolling, but supporting the process. They were only in the supporting system. They were not involved in their way of protecting or managing the forest. Gradually, neighboring villages started joining the initiative, and by 1995, a total 95 villages became part of the federation. The patrolling squad was very effective in combating timber smuggling in the area. Since there was a lot of timber smuggling and there was a lot of forest was cut down due to timber, so with the patrolling system, with the formation of the committee, there were the patrolling system started controlling over the timber smugglers. Then consequently, in 1992, its strength then was reduced to 21 members, including three women. So there were also women protectors as well who were part of the committee. Now, Budi Khamari as a sustainable food security. In what happened in post protection of the committee. So in first few years of protection, the availability of sal and tendu leaves rose markedly. Seed production has been improved. We're seeing the seed production was gradually increased. were improved in 1992. In 1992, there was it was found that 78 tons of sal seeds were collected by generating an income of rupees 97,500. Similarly, production of orange seeds and mahua flowers was also increased. Prior to the protection, a number of villagers were engaged in fuel wood collections and charcoal making. So how you can see there, it was actually helping them, the entire system helped them to generate their livelihood and food security. These villages have now switched to collection of sabai grass for rope making as an alternative occupation as well. So there are sabai grass as well, which was again become a another mode of livelihood. According to the residents of Budi Kamari village, the regenerating forest provided some kind of employment to every villager. So as I told, every villager has a role to play in protecting the forest. So they used to go for patrolling, and let me also tell you one thing, the committee collects fund to give a daily wage, losing a daily wage. So when a person is actually looking after the forest, he or she use a daily wage. So this committee, they collect funds and they pay the wage of that particular day. Villagers are also free to collect the non-timber forest produce. Besides, the agriculture production seems to have increased because of increased availability of water, increased fertility of land due to reduced soil erosion, availability of fodder and fuel is also much higher than before. So you can see how gradually the forest improved and their lifestyle as well. So with this, I also have something to add. A conference which was organized on forest for food security and nutrition was held at headquarters in Rome in May 2013, conveyed that the forest, trees and agroforestry system demand greater attention in strategy for food security and nutrition and in the fight against the hunger. As I said, food security is first agenda of Millennium Development Goal. It also called for improved data collection at national and international level. So let me now show you who are these Budi Kamari forest protectors. I have a small video to show. समझतेगा?
आशा बंधु जुड़ो हाथ नाच लगाई दी बोए आशा बंधु जुड़ो हाथ नाच लगाई दी बोए लाल माटी साल बोल जुमाई दिलो अंधेर मोन लाल माटी साल बोल उड़ाई दिलो अंधेर मोन आशा बने उड़े आका बोरे आशा बंधु जुड़ो हाथ नाच लगाई दी बोरे आशा बंधु नाच लगाई दी बोरे
So this is a small clip which I documented during my work in Budi Kamari. So as you can see how happy these women are, protecting their forest, and you can see a lot of fuel wood on their hand, and there were also women who were collecting MFP from the forest. So how it actually regenerate the forest and as well as the food security. With this, I end my presentation. Thank you very much.
Now, we'll start the third one. Now I request Dr. B.K. Bindhani, Professor KIT, Odisha. Now he is going to deliver the present and future role to sustainable agriculture systems through genetically engineered crops for food security.
Yeah, good afternoon to all. And respected chairpersons, M. Pushti, co-chairperson C. Kubera, and rapporteurs Dr. C. Mandal, and all management staffs, and also the respected BC sir, Den Rao. And it is a great privilege for me to inviting for this national seminar on sustainable agriculture and food security. Thereby, thanks to the management committee and also the Centurion University. Let me start my topic, that is the what present and future role of sustainable agriculture systems through genetically engineered crops for worldwide food security. My dear students, you should attentively listen how the genetically modified foods will be formed and how it will be having all merits and also demerits. First of all, I am describing here what are the merits. Why will go for the GMO, genetically modified organism, or genetically engineered organism, GO? Why? First you see here, why? Because nowadays that is the most common in the agriculture sectors, that herbicides and also pesticides. So that will be developed the varieties. There may be herbicide resistance plants, insect resistance plant, or you may say the disease resistance plants, transgenic plants. We know that all. That is also transgenic plant through the, that is also GMO also, it is otherwise called as transgenic plants. Because the plants, normal plants and the transgenic plants, only basic, there is the what, the normal plants having the basic or parental DNA, but in transgenic plant is the foreign DNA. How to make it? We'll discuss. And frost protections, that is the what, so many plants are going to be extincted. So how to conserve, germplasm conservations, that is called cryopreservations, means preserving what in frost or frozen state. There are so many methods having, that is the what to keep it in frozen state. Basically that is the liquid nitrogen, mostly used, that is minus 196 degrees centigrade. Like this, there are so many also having the used for the cryopreservations, or that is the frost protections for the germplasm conservations. Longer storage life, that is the most important in agriculture sector. Then new crops productions, only technique by which we can produce quickly the new varieties, having some novel characteristic features. Flavor, the most important. Nutritional varieties, having the more nutrition, shelf life of products, and any other property of the plant that improves its value. This is, you see, the product, golden rice. All you have heard it, but not available in the markets. Still it is in research. This is, you see, the rice, varieties. Golden rice contain increased level of provitamin A. Traditional rice, that is white, you see, and that is B is the yellow rice, that is golden rice, was developed in 2000s, and is light yellow color, that is only 1.6 mg per gram of the carotenoids. But 2005, you see, that is the transgenic lines are developed, that is dramatically increase the amount of keratin synthesized, that you see the last one, making the rice is deep golden color. That is the 37 mg per gram, that is the what, carotenoids, but it was earlier only 1.6, which 84% in beta carotene trial, also going on the research. You see, this is the what, maize bana, that is also products of the what, GMO. This is, you see here, the firefly, fire tobacco. These are the what you see here, the tomato comes in many varieties color. These are the transgenic tomatoes you see here for food security agriculture sectors. Expressing different malaria antigens. This is, you see, that the delivery of the corn-based edible vaccines. That is transgenic corn snacks, embryo germ cells you see here. These are the, you see, tearless onion, that we know that they having the onions are sliced. That the what, that you can market, everybody will be tear. But now the GMO, that is the what, a tearless onion, the research is going on. This is the, you see, this is the orange purple green cauliflowers, developed by the what, GMO. This is the what, the first blue rose, developed by the transgenic, that is GMO. You see here. You see here now, what, when, why, how, when, that is the which, where, that is genetically modified organism. What is that? Now we'll see. Genetic engineering, that is G, focused on the manipulation of DNA. We know everything is DNA. By scrambling or blocking or adding genetic materials. Or simply we can say it, my dear students, genetic engineering means there is a designing of genes. A scientist can design a genes like a civil engineer or architect engineer construct a home by using the bricks, cements, etc. Like this. the genetic engineering, basically, that we can compare with the scientists or so many scientists all over the world, they're designing the gene according to their requirements. That's right. That is the result has been dubbed a genetically modified organism or GMO or GEO. You see here, genetic engineering, making the engine by using laws of genetics. We know everything, that is the Mendel's principles. Genetic material, the total composition of the genome, genetic material of any organism, the complete genome, controls in inheritance and the phenotypic expression. Phenotypic, we have all known that the Mendel monohybrid cross, the F1 generations, the expression, there is a two genes, that is phenotypic. Genetics, that is what? Genotypic also. Means suppose F2, there is a what? The ratio is that the phenotypic 3:1, but the genotypic ratio is 1:2:1, like this. So phenotypic expression, that is the, we can observe it. You see here, phenotypic, one example you see here. Phenotypic. You see here, the human race machines, that is Albert Einstein, you see. These are the phenotypics. This is everything controlled by the DNA, you see here. So what are GMOs? Now question, what is genetic engineering? We have already known. Now what is GMO? Any organism in which genetic material has been altered in a way that does not occur naturally by mating, that they already been told that the hybridization not. That is what? Genetical materials and natural recombination manipulations. Or in principle, genes may be species could be inserted in any other species. So basically what happened there? Combination of different genes of the different species to form a hybrid combinations, DNA. Combination of DNA segments from the different species to form a what? Recombinant DNA. The phenomenon is called recombinant DNA technology. All we have known it. So you see here one example. First one you see, this is the what? Carrot. All you have seen. Scientists to copy carrot gene convert a pigment to beta carotene. How there is the next step you see? They insert the carrot gene into a plasmid because we know that genetic engineering or recombinant DNA technology, 4 important tools. First one is a restriction enzyme or endonuclease. That will be cut, biological seizure. You can cut it, particular segment of the DNA to make it a sticky and blunt ends. And how? That is the what? Next is what? Carrier vectors. Like this we are keeping the bags. Like this is the what? Bags. Carriers or vectors. Plasmids generally used. Then we can go for the what? Ligations, where the broken part can be joined or cut part. Then host cell. Like this, there is a carrier, there is a what? The genes, we want to convert it. That is the what? They insert the carrot genes into the plasmid. Then the plasmid is introduced into the agrobacterium. Generally, in plants, in agriculture, we develop the plants, transgenic plants or GMO, whatever maybe you are telling that the what? Through the agrobacterium transformations. That is the TI plasmids and RI plasmids. Tumor inducing and that is root inducing. That is agrobacterium tumefaciens and agrobacterium rhizogenesis. Generally, we methods. We will discuss again how it will form. Then plasmids, the agrobacterium transfer the carrot gene to the cells of the tomato leaves in petri dish you see here. Sorry. You see. Then this is the tomato cells grown and divide into a culture with the hormones that encourage the cells of the become new shoots and roots. And here you see, this is the age that the new plants grow the carrot genes, convert the tomato pigments. What happening? Into beta carotene, creating an enhanced tomato you see here, plants. That is the GMO. So this is the, you see, transgenic processes, you see here, engineering, technology, use of knowledge of or mechanical art and applied science of for humanity. This is the technology. Then engineering, creations of engines, application of science to design device and use of machines. And next is engine, mechanical device of different parts working together. That is engineering. So engineering streams, all having this is also management engineering institutions. These are the stream, also others also, etc. But here, engineering and genetics I'm comparing, you see here. Engineering coordinations of the principles, but genetics, universal principle of working. And genes, intrinsic capacity of an organisms. And genome, gene, DNA, or nitrogen bases, we know that. My dear students, you know what is the DNA. Nitrogen bases, gene, DNA, you saw all you have known. Universally same, the microbes, plants, and all things. This is the DNA, nitrogen bases, universally occur, same things. Here you see universality. This is the components, DNA you see here. The the purines and that is pyrimidines, bases you see here. These are the replications, DNA. Then this is the expressions you see here. And this is the regulations. This is the regulations. The engineering coordination, we have already discussed. Since the last 25 years, genetics become technology. Now that is the genetics, that is the technology. Manipulating genes for desired behavior of living organism like a machine. That I have compared engineers and also genetics. Methodology you see here. First, that is the targeting host and desired traits. Gene isolations, gene preparations, cloning into vectors, introduction into host cells, then selection of transport cells, then characterizations of the GMO. These are the methodology. First, targeting or desired traits, what we want actually in plants. That I have already shown you how the carrots form the high-yielding tomato having the more carotenoids. Like this rice, so many varieties I have shown you. Gene isolations, so many methods employed to isolate. Gene preparations, then cloning into vectors, the introduction to host cells. Generally, host cells, that the bacterium will be used, E. coli. Selections of the transport cells. Then we go for the characterization, so many methods by which we can characterize. Molecular methods. So tools, gene isolation, molecular seizures, that I've already told you, that the endonuclease, gene amplification, cloning PCRs, carriers, that is vector, I have told you, plasmids, so many vectors, natural and artificial, I will show you, gene transfers, identification, morphology, and markers. So you see here, this is the molecular seizures, that is the, I have already told you, restriction enzymes, so many restriction enzymes. You see here, these are the Nathan, Smith, and Arbar, that is that what constructed the first genetic map using restriction enzymes. Then this is the purified one restriction enzymes. Then the postulates, that is the big scientists you see, they were awarded by the Nobel Prize, that Albert Nobels, they were awarded. That I have already shown you. Restriction modification systems, defense systems, restriction modification systems. Why will go for the modifications? Then a pathogen entering any living system can be defended through restriction systems. Any pathogens can be controlled by the defense system, by the modification systems. Own genome is protected by modification genome, by DNA methylations. And this is, you see, types, restriction enzymes. We know that EcoR1, ECO2, HIND3, like this. There are so many restriction enzymes. Time is very short, so I have to finish it quickly. So these are restriction enzymes, how they cut and the particular segment of the DNA, particular segment, particular selected segments. And these are the restriction enzymes, not required. And restriction endonuclease, type 1, type 2, type 3. There's a type 1 complex endonuclease, recognition sequence, that it will be base pairs, it will be varied. Type 2 also remarkable stable, recognition that will also vary from the base pairs. These are also enzymes, restriction enzymes. Type 3, that is the intermediate types, cleavage DNA intermediate vicinity or R, site functionals. These are examples I have given. And restriction sites, recognition sequence, that is palindromic sequence. There is the 5 days and 3 days, the DNA sequence, you know it. There may be palindrome with rotational symmetry also. And they are also like this, the GAA, TTC, these are the G means guanine, adenine, like this. These are we know that from the DNA, nitrogenous bases. These are the tables showing the community used the restriction enzymes and where that will be obtained. All the restriction enzymes, so far there are 275 restriction enzymes discovered, and all are obtained from the different types of the microbes and named according to their origin of the microbes. They are written here, you see here, you can see here. And these are the blunt and how after applying the restriction enzymes. And this is the principles, human DNA, EcoR1 sites, suppose DNA and plasmid vectors you see here. These are the plasmid vectors, EcoR1s, and origin of the replications, then cleavage. When you will be apply the restriction enzymes, you will cut into make it small pieces. So it will carry by the plasmids you see here. You can also apply the restriction enzymes, then joint it and to form the new varieties. Like these tools, genetic isolation and molecular scissor, we have already discussed these ones. Then gene amplifications, gene cloning. These are the PCR, general done. They're done by the Karimulis and also awarded by the Nobel Prize in the year 1993 for these inventions. You see here, clonings. This is the dancing after getting the Nobel Prize. And PCR you see here, detection of the chromosome translocation, amplification across the translation sequence, chromosome painting, detection of residuals, like this, so many having a small chromosomes, we can, or DNA, we can, more thousands, billions, and billions number we can produce. And then carrier vectors, external vectors, there are so many vectors I was telling, that is the cloning vectors and also expression vectors. These are the properties of the good vectors. Why will select the vectors should have the properties, autonomous replications, AG isolation of the purification before selecting the good vectors. We should care or we should take the some properties. Suitable markers, integration protein with the DNA inserts, restriction sites, control elements, and promoter and operator. These are the the vectors types, natural and artificials, plasmids, phages, virus, transpose, and these are the artificial plasmids, phagemids, shuttles, and replacements, artificial and retriever. These are the vectors. So single copy, one copy of the multiple you see, 10 to 20 copies partial you see after the insert of plasmids. This is the plasmid you see here, bacteriums. And these are the plasmid types, F types, F plasmids, the conjugations, generally bacterial conjugation, all you have read it. R plasmids, restriction genes, and call plasmids, that is immune proteins. This is synthetic, that is what our antibiotics will be released and will kill the cells. These are the call plasmids, immune protections. Plasmid protections, the minimum amount of DNA, properties, relux replications, at least 2 selectable markers, multiple recognition sites, R site within genes, and these are the examples. You see, these are the example plasmids, that is the PBR322, that you see the sites, labeling properly here. And this is plasmid PBR322 map, you see here. And these are the bacteriophages, phages lambda, phage M, that is the word extensively used for the vectors, phages. These are the phages lambda vectors. You see here, this one. And this is the phage lambda vectors. These are the Catt's features. And cosmids, this is also artificials, plasmids, all should carry the selected part of the DNA. These are the cosmids, you see here. That the the ampicillin, tetramycin, these are the tested for the characterizations, the antibiotics. Recombinant DNA you see, constructs RDNA, recombinant vectors. Gene source, living organisms, gene machines, gene library, genomic DNA library, or cDNA library you see here. Genomic DNA library. Genomic DNA library you see, extraction of entire genome DNA, restriction, digestion, simple or mixed. Then this how to genomic DNA library constructs. First extraction of entire genome DNA, then restriction, digestions by endonuclease, separation of fragments of equal size, then insertion of fragment in vectors, then vector containing genome DNA clone in host. Then genomic DNA library, mechanical sharing, sonication, you see the methods mechanical. Then this is the sonications, and this is the restriction enzyme to make the genomic DNA library. So electrophoresis, separation of fragments, equal size, how can done it? Electrophoresis, density gradient centrifugations, and these are the genomic DNA library. Organisms, genome size and fragment, E. coli, yeast, Drosophila, and human. You see here, genome size and fragments. And this is a genome DNA library application, genome conservation, ready availability, multiplication of genes, source of the genes. And these are the complementary cDNA library. It is a good library consists of only those regions of the genome which are expressed that construct from the mRNA. And this is another part, how to make it, you see here, 5' and 3' or 5' you see mRNA, primer with oligo DT, reverse transactions, reverse transcription, how to form you see here. Then mRNA DNA hybrids, you see here. And RNA degrading, that is alkali mediums, degradation, then single stranded DNA with loop, then the DNA polymerations, and then double stranded DNA with loop, you see here. How to form this? See our complementary DNA library, that is the double stranded DNA library. And then SI nuclease cleavage hook, then double stranded DNA. Then you see here vector cloning, then a recombinant vector with cDNA placed in bacteria. Applications. Only these genes are preserved. Smaller than the genomic DNA library, intron less than structures. As RNAs are small size, it can place the normal vectors and expression vector, cloning vectors. Applications. Then limitations, heterogeneous time of gene expression with resides. All genes are never direct actions with any times. And that is the processed DNA is used, hence no similar sequence of the DNA. These are the limitations. And RNAs are having very short lifespan. And this cDNA have blunt-ended, hence requires linkers of adapted DNA molecules. These are the limitations. The selection of the transformants. These are the morphological observation, tagging, antibiotic resistance, ampicillin or tetracycline, marker gene, FGP or LUX, these are the enzymes. There is the luciferase and the green fluorescent proteins, electrophoresis, hybridizations and chromatography. There are so many techniques. And the tools you see here, gene transfers, you see here tools. There are so many techniques. That is the what? Generally, there are the two main types of the transformation. One is direct methods or vectorless methods. Another is indirect methods or that is vector-mediated gene transfer. Generally, these are the what? That is the vectorless or direct methods, chemical method, electrophoresis, particle gone method, gene gone. Generally used. Direct, that is the direct methods. Microinjection, macroinjection, pollen transpiration, these are the methods. And these are the what? For the RI and TI plasmids. You see here, this is the what? The agrobacterium. You see agrobacterium, there is a gene gun. Gene gun shows the gold particles coated with the DNA, and there is a gene inserted into TI plasmids, tumor-inducing plasmids. Bacterium mist. How it will be inserted? Details I will show you, and then it will get into. But there is agrobacterium. Through also you can insert the gene, desired gene. Here also there is a gene application. There is a gene gun methods. Also there is a gun particles coated DNA by which cell sort into genes gun and DNA incorporated into the plant cell. And you see here the chromosome with the foreign DNA. Here it will be what? You have to select it, cell screening. Where that is you see here clearly marked the foreign gene inserted in the chromosome. So it is, you can say it that is transgenic plant. By that methods we can develop the transgenic plants. Either maybe agrobacterium or gene gun. You see, this is gene gun machines. Vectors coated into golden transient particles. Particles are selected high speed by the gun. Particles enter plant tissues. Then enter the nucleus and incorporate into chromosomes. And this is the what? The laboratory model, and this is the garden model. Now it will be used, lab versions, portable version also available for insert the genes to get it transgenic plants. These are the gene guns, principles, you can read it. And that I have already discussed, agrobacterium mediated. This is agrobacterium mediated gene transfer. That is the vector mediators. We have already discussed vector mediated. That is the what? Agrobacterium. These are the what? Gram-negative soil bacterium. How it will be formed? To form the tumor inducing. That you see plant pathogen found in nature. That is found in every nature, anywhere else. And how it will be introduced? You see here, fungal stems go on the leaves. And these are the gram-negative, I have already told you. And it will be what? Formation results of the transfer of integrated expressions and specific segments of the agrobacterium tuberculins, plasmid DNA called the tDNA or transferred DNA. And the tDNA is, resides on the large plasmids called the TI or tumor-inducing plasmids found in agrobacterium tuberculins. This is you see here, the galls can be huge, fungals. These are the what you see here, fungols, that is the what? This is the method agrobacterium tuberculins. Where genes TI plasmid TNA you see here, how it will be inserted in the plant cell you see here. This is the process by which it will be inserted to the what? Plant DNA. Here you see already been transferred, you can mark in red colors. So that plants will be transgenics. This is the methods by which you can develop the plants. This is also methods by which productions of the transgenic plants, you see here, through the agrobacterium transformations. And TI plasmids, tumor instrument, not necessary to discuss again. So these are the construction of the TI plasmids, tDNA, transfer DNA. So you can read it. These are the constructions. RI plasmid, root inducings. These are the, you see here, methods to produce lactoferrin strawberry. This is the cow, you see, this is the lactoferrin gene. These are the what transport into the agrobacterium. This is the what strawberry leaves, you see here cut. And these are the strawberry, that is the what containing lactoferrin, you see here, and dipped into the transfer and to call the medium and develop into the plants, you see here, strawberry. Having the strawberry containing lactoferrin. This is collected from the cow. This is the what method of producing lactoferrin in strawberry. Like this, you see some news, we are using GM, genetically modified gods, to make human breast milk, says the Russians. These are the news you see here, recent, published in the net. This is the what stop bill on genetically modified crops. The earlier respected our Honorable Prime Minister Manmohan Singh has told that is the what stop bill on genetically modified crops. NGO tells Manmohan Singh, you see here, Honorable Prime Minister. But nowadays, this is the Nature Biotechnology published. This is the genetically modified crops. The demand, it is increasing. Year by year, it is increasing. You see the graph increasing because it has so many merits. Other than the less demerits, and by which we can, or the sustainable agriculture and food security problem can be solved, not only in India, also all over the world. Thank you all.
Senior scientist CRRI. He will deliver the lecture on role of microbes in sustainable agriculture. This is the last topic before lunch break.
Good afternoon, one and all. Without taking too much of time, I directly enter into slides. At the outset, I really thankful to Dr. K.K. Behra, who has invited me to deliver a talk relevant to the national seminar theme, Sustainable Agriculture and Food Security. My topic is my beneficial microbe, a sustainable solution for food security. So I am focusing on these 5 aspects of microbes. One is how the microbes are interrelated with the plant in terms of carbon source, nitrogen and phosphorus source, and how plant are giving the nutrient source, and in return how microbes are giving to the plant. Another thing, the biocontrol and biofertilizer agents, because the microbial resources are huge and it is utilized as a biofertilizer and biocontrol agents. Then what development so far achieved and what gaps are there, I am going to discuss in detail. Third is, what is the effect of inorganic and organic fertilizer in long run? Why? They are actually affecting on soil microbial community in a long run. The microbial community is going to down, or they are showing the same diversity as earlier. Another thing, then if climate change is there, so if elevated temperature is there, elevated carbon dioxide is there, likewise, then what is happening to the soil microbe? What is happening the status of soil microbial community? Then another thing is the microbes, which is extreme tolerant, extremophiles, how they are going to mitigate the abiotic stress which is arrived due to climate change. The first aspect is the plant microbe interaction. We have legumes, cereals and oilseed crops, generally brassica. So there means the legume having all crops are autotrophs, means they are taking carbon from atmosphere, carbon dioxide, and the photosynthesize, and in return in photosynthesis they are converted into glucose, means they are autotrophic, they are generating their self the carbon resource. Then how they are dependent upon the nitrogen. If you take as the pulse crop means how their nitrogen requirement is fulfilled, it is due to nodulation, due to rhizobia. So what happened? The rhizobia, which is associated with the legume crop, it is actually fixing the atmospheric nitrogen and reduce into the ammonia. So what happened? This rhizobia is dependent upon the plant. Carbon source is supplied by the plant. In return, it is giving to the nitrogen. So the symbiotic relationship is there. Now come to the phosphorus. So here also arbuscular mycorrhiza is there. I think you all aware about arbuscular mycorrhiza. So what they are doing, the phosphorus which is present in the soil in unavailable form, it makes it available and the mobilized into the plant. So, and it is dependent upon the plant, so carbon sources are coming from the plant, and as a rent they are paying the phosphorus. In legume, in nutshell we can say that it is a complete symbiotic pathway is there. Means carbon sources are coming from plant, in return nitrogen and phosphorus from different microbes. But you look at the cereal. Cereal means they are supplying the carbon source, but their nitrogen fixing bacteria are not there. Have you seen any nodulation in cereal plant? Rice, wheat and all those things? No. But the attempt are going on. Some scientists develop the nodulation, that is a paranodulation, but it not recovered. So what happened? Legume means symbiotic pathway is complete. So scientists are trying to transform all the symbiotic pathway into the cereal. So what happened? This cereal plant is wholly dependent on the chemical fertilizers. We apply urea and all those things. So what happened? Ultimately, soil is deteriorating, soil health is deteriorating. So if the nitrogen fixing bacteria is incorporated into cereals, then it is a big achievement. Likewise, here arbuscular mycorrhiza is there, but it is not so much feasible. It is mobilizing the phosphorus, but the major concern is nitrogen. So nitrogen fixing bacteria is another. Other bacteria is there, like as endosymbiotic bacteria is there, but their activity is unknown. Scientists are trying to success whether they having the nitrogen fixing ability or not, because they are like rhizobia, they are also dependent upon the carbon sources from the plant. They are leaving the tissue of root, leaf and stem, and they are utilizing the carbon source from plant. But in return, they are giving any nutrient like nitrogen or phosphorus. This is the actually gap. We are just going to solve it. Brassica, there is no symbiotic pathway. They are rudimentary. They are highly dependent upon the chemical fertilizers. There is no symbiotic relationship, because in Brassica, like rapeseed mustard, the root exudation having sulfur compound. So due to this sulfur compound, the association is not there. No nodulation, no arbuscular mycorrhiza. Here the research gap is more. So we have to develop such a way so that the nitrogen fixing, the all symbiotic pathway should be transformed in the Brassica to fulfill. So these are the major constraints we have to do to solve this. Coming to biofertilizer. So here I told you already, this in cereal, nitrogen endosymbiotes are there as a nitrogen fixing. What achievement we did? What in one study which I did, means I have isolated some endophytes which is actually nitrogen fixing bacteria. So how we will confirm? So it is confirming through basic research, and then you are going to strategic, and after that in farmer field as a applied. So first means we have to go for basic. Basic means how can you know the bacteria, the concerned bacteria is actually fixing the nitrogen. So we are isolating, first means we are isolating the bacteria from root or different regions. Then you are going for nitrogen free media. If they are growing, means tentatively we can say that this is nitrogen fixing bacteria. Then we doing the acetylene reduction assay, because the nitrogenase is the enzyme which is actually play important role to fix the ammonia, fix the atmospheric hydrogen and finally reduce into ammonia. So endosymbiotic bacteria having nitrogenase enzyme or not, so this NIPase gene, actually NIPase gene codes the nitrogenase enzyme. See, the nitrogenase enzyme having two components, the component 1 and component 2. Component having two MOAP protein and component 2 is Fe protein. Component 1 is MOAP protein having another two subunits, that is alpha and beta. The alpha component is coded by NIPDA alpha beta component is coded by NIPK, and the component 2 having Fe protein which is coded by NIPH gene, and that is very important because in reduction atmospheric nitrogen which is reduced in ammonia, the first electron is accepted by NIPH. If NIPH is there, means that confirms, means that bacteria having nitrogenase enzyme. And we can see here the gel, means the bacteria which is showing the this fluorescent structure, fluorescence under gel condition, means this is showing the, they having the NIPH gene along with azoospermolum. And this showing the diversity, means we conclude that we have certain endosymbionts in right tissue, rice tissue, aromatic rice tissue, which is going to fix the nitrogen. Then another means we also work on 3 cultivated rice variety and 1 wild. 3 cultivated Sabita, Swarna, Swarna Saban, and 1 wild Oryza echengeri. We isolated, and that uncultured bacteria which is not going to cultivate in media. So what happened? We are directly seeing the DGE, and this diversity, what it is showing that means they have different types of bacteria living inside the tissue which shows their NIPH activity, nitrogenase activity. After that, what I told, then you move on, means basic to applied. Whether this endosymbiont which we have isolated from rice tissue, they are working well under field condition or not. Then what we did it in field condition along with control, recommended fertilizer dose, and half nitrogen plus nitrogen fixing bacteria, half phosphorus and phosphorus, and we come to this conclusion, then they are working well under field condition. Coming to another aspect, biocontrol, means bacteria or microbes, they are having antibiotic. See, in this rice field or agriculture crop condition, we are, spraying the chemical pesticide, which is actually deteriorating the soil health, plant health, many more. So why not you use the microbes, which is actually secreting the antibiotics, which kills the pathogen and pest, because due to for sustainability, the microbes is an alternative. So what happened? Some biocontrol agents are there, like Pseudomonas fluorescens. So we have isolated from the rhizosphere and tissue of rapeseed mustard, Brassica junicea. So what happened? The fluorescent Pseudomonas actually secreting 3 types of antibiotics. One is DAPG, phenazine, pyrrolutrin. So again, we have to confirm whether the antibiotics they are secreting or not. So certain genes are there for respective antibiotics. So we amplify along with control. Control means the bacteria which is already identified. This means that bacteria is actually producing the antibiotic. So we confirm with this PCR, and after that TLC and dot blot hybridization. Then if you are finally sure we have antibiotic gene, then again go for field condition, field or pot condition to reconfirm that. So here also you see the 3 bacteria we isolated from the rice, and against it is acting against the seed blight, and it is confirming that this our isolates along with positive and negative control, they are doing at par with the Trichoderma viride and even chemical control. Likewise, our bacteria is also as effective as this chemical and the biological control to inhibit the bacterial leaf blight of rice, like Xanthomonas oryzae. Another aspect is coming. We are using exogenously the nitrogenous fertilizer, phosphorus, and all that. In CRRI, the one experiment has been set up, that is set up in 1969. So what means here I would like to demonstrate that after 43 of a year of experiment, what is happening of microbial community if you exogenously use the chemical fertilizer along with organic fertilizer. So what? There the different treatments are there, and you can see clearly how the microbial community are diminishing if you use nitrogen fertilizer without using any organic. But you see here, if you use nitrogen fertilizer along with farmer yard manure, their microbial community is rich rather than only nitrogen fertilizer. It is due to many reasons, but one of the reason is if you use nitrogen fertilizer alone, what happens? The pH is going to down. Okay, so to supplement this pH to neutrality, okay, what happens? FYM is acting as well. So, and here you see the AWCD is average well color development. I mean, this is a biolog based microbial community, okay, and there this is a diversity index like MAC index and Sun index, and it is showing here also. See, FYM plus N means combination of inorganic fertilizer with organic fertilizer. Actually, the doing well means it maintains the microbial diversity community rather than if you apply the nitrogen fertilizer. One interesting report, this all are my report because this is this recently means I developed, okay, and this all are under review means I communicated also. One thing also where the means bacteria having significant relation with antioxidant enzyme. So this is actually showing the correlation of bacteria with antioxidant. Means we did with actinomycetes, dehydrogenase enzyme, fungi, fluorescent diestate, heterotrophic aerobic bacteria, MBC. Means out of these, the actinomycetes and FDA both are showing the correlation with antioxidant enzyme. Means if this one is increased, means definitely antioxidant enzyme are increased. Another part is the burning topic. Then what is the effect of climate change on soil microbial community? Okay, and I'd like to mention that if soil microbial community is more, means this indicate that means soil health is there. It is a good, means life part of soil is life part of soil microbes, and this year also celebrated as International Year of Soil. See, even once again repeat, this year is 2015 is regarded as International Year of Soil. Okay. Now see, the climate means climate change having direct or indirect effect. Direct effect is due to temperature and moisture combination. Then what scenario having? Two type of changes is there. One is modifying the physiology of existing microbial population and changing the structure of microbial combination. Means direct effect due to moisture and temperature, two is there. Means either it is just modifying the physiology, means respiration rate is high, respiration rate is low, and the changing of the structure. Maybe it become gram positive to gram negative here. Maybe fungi become actinomycetes like this. So indirect effect means elevated carbon dioxide. Elevated means elevated carbon dioxide showing the cascading effect on plant metabolism, growth and diversity. And it also changes the soil physical property like soil moisture and resource quality. Based on this, Singh et al. 2012 published a paper in Nature, and they hypothesized that if the global change is there, okay, global change is there, and the soil process, state 1 converted to state 2, then what happened to soil biota? The first hypothesis is saying that, okay, this is the current condition and this becomes this, and this is due to physiological activity. Means they are changing the metabolic activity. Suppose gram-negative bacteria is there, if temperature increases, so sporulation is there because temperature is increased, so maybe gram-negative bacteria is withstanding the temperature, okay, modify their physiological needs, but this one is not converted into gram-positive bacteria. But another way, another hypothesis is their community is changed. Means if harsh condition is there, okay, more than 100 degrees centigrade is there in hot spring or something, so what happened? Gram-negative bacteria is entirely converted to gram-positive bacteria. Means their community is changed. They're going to dormant phase. Then what happens to elevated carbon dioxide? Elevated carbon dioxide, if it is there, so actually what happened? Plant is taking the carbon dioxide photosynthesis. After that, it is secretion is there, okay? Secretion is there through root. So if elevated carbon dioxide there, then through root, plant root, it releases of labile sugar, amino acid and organic acid. So what happens? It stimulates the microbial growth and activity. If microbial growth is more, then what happens? Microbial biomass carbon increases in the long run. If microbial biomass carbon is there, then what happens? It is mobilize the nitrogen for their growth and development. So nitrogen is limiting to the plant because nitrogen is not available to the rhizosphere. So what happened? Plant is diminishing. But in another case, the CN, it becomes the negative effect of the plant growth. But here nitrogen is less, then CN ratio increases. So in this condition, it favors the higher fungal dominance and diversity rather than bacteria. So higher C assimilation and lower decomposition of cell wall by fungi than bacteria. Fungi, the cell wall is made of chitin, and there means bacteria is N-acetyl glucosamine, okay, N-acetyl muramic acid, that is easily decomposed, but chitin is not easily decomposed, but then higher carbon sequestration is there. Likewise, temperature. As we know, the average global temperature is predicted to increase by between 1.1 to 6.4 degrees centigrade by 2100. So what happened? In this condition, the heterotrophic microbial activity increase the respiration, and the 100 million tons of carbon released due to, if 2 degree temperature is increased, then 100 million mainly due to microbial activity. Okay? See here. This is showing how the microbial group, how the microbial activity are changing due to elevated carbon dioxide and elevated temperature. This is the well predicted with elevated carbon dioxide and temperature. Here it is not there, but this data is actually showing if elevated carbon dioxide and is there. Okay? So it is showing the significant relation with the microbial biomass carbon and soil enzyme activity. Okay? Moisture. If microbial community and the decomposition of the soil organism is affected by, if 20% increase or decrease of soil moisture is there, there is significant decrease or increase of microbial community is recorded. See, if soil moisture is there, it is directly respond because water is required for physiological activity, and if larger period of dry condition is there, means dry condition means water is not there. So what happened? It limits the microbial growth and decomposition, so it shows the negative impact. Likewise, indirect response is there. So if wet condition, the gas diffusion rate, oxygen availability is there. In that condition, soil drying may increase oxygen availability and hence the carbon cycling in wet. So it's showing the positive feedback. So there is balance of negative and positive feedback of climate change. See here, atmospheric carbon dioxide is there. Through photosynthesis, carbon dioxide is converted into glucose, and plant again release the organic carbon and sugars. Under anoxic condition, suppose rice condition is there, which is actually flooded. Under anaerobic condition means there oxygen is not there. In this condition, this organic compound is utilized as a carbon source by methanogen and converted into methane. Okay, anoxic condition I must say. So what happened? If flooding condition reduced, then another bacteria come into picture that is called methanotrap, and this methanogen is utilized as a carbon source by methanotrophs and converted back into carbon dioxide. So balance is there. If flooding condition always there means this world are saying that India is contributing more emission. Why not China? Why not someone? Means rice cultivated in more means more methane emission. This is not there because flooding, after flooding, certain dry period is there. If certain dry period is there, then methanogen is come over by balancing by the methanotrophs. Once again, anoxic condition is there. This organic compound is converted into methane by methanogenesis, and if dry condition is there, then this methane is utilized as a carbon substitute by methanotrophs, then it is coming back to the carbon dioxide. Okay, then balance activity is there. Two type of methanotroph. Methanotroph means utilizing methane as a carbon source. Two types. One is low affinity, another is high affinity. Means low affinity means active only. Means they are not so much sensitive. If more than 40 ppm is there, then it will become active. This is also called type 1 methanotrophs and which mainly belong to class gamma proteobacter and often consume large proportion of CHE products. Second is the high affinity, means very sensitive, means if less than 12 ppm also there, they consume methane, and this belong to the alpha proteobacter and remove approximately 30 million tons of methane. If warmer condition is there, okay, warmer condition means it means in terms of methane I am telling you, if warmer condition is there, it could promote oxygen availability. If oxygen availability is there, methane emission is going to down. Elevated carbon dioxide also affect the methane emission indirectly through their effect on microbial activity. Okay, there are three concepts. What I am saying, the concept. One thing, if plant mediated moisture is there, means water is there, in that condition increase methane emission. Water is there means anoxic condition, methane emission. So here increasing methanogenesis than methanotropy. Means I told you the balance, methanogen and methanotroph. So here plant moisture is there, then methanogenesis increase than methanotroph. If higher temperature, then moisture is reduced. The reverse is the case. Here methanotrophs are more than methanogen. If increased precipitation means rain and temperature, just like a rice ecosystem. So what happens? This type 2 methanotroph जो है decline, it is going to decline. This is a example, very good example of novel research. Novel research means new idea. So what happened? Means one alternative way by Pramanik and Kim. This is published in Geoderma, very reputed international journal. What they did? They apply EDTA in pot experiment. And this EDTA actually having the one component, cobalt. It is actually what happened, this is chelating the cobalt, and cobalt is the cofactor for methanogenesis enzyme. Okay, so what it reduces the methanogen, and in turn methane emission is reduced if you apply this, means this much of EDTA. This is a basic concept he used and he published. So this is overall picture of climate variable, and it is interdependent upon the biological variables. Now I told you the last slide means some extreme tolerant microbes are there which is going to mitigate the stress condition. So certain extreme tolerant microbes are there which is showing the plant growth promoting traits, and this picture it is showing that this is in elevated temperature and this is the higher temperature. You can see the plant growth promotion is there still in elevated temperature. But if you look at the elevated temperature, see bacteria primed seed is showing plant growth promotion than control. Means it is actually mitigating the stress. Likewise, this Naveen variety of rice is salt sensitive variety. Even 2 decimon if you will give, then it will die. But our bacteria, if our bacteria has given, then it is giving, okay, the growth. Means the extreme tolerant, means salt tolerant bacteria is there, and it also induce. But you see this is without salt, it is increased so much. So it is actually enhancing this not yield, but the parameter of root length and something, but not like control. But if you see the control of salt, means 2 decimon without bacteria, there is no shoot at all. So overall, I would like to say how to assess the microbial community. If certain condition you have, then how to assess. So this is the one sort we can do. If you do this all, then you can analyze all microbial community. First means culture dependent, means you just assess the structural diversity. Structural diversity means you count. Means in this condition, how many bacteria, fungal, actinomycetes, heterotroph, sulfur producing bacteria like it is there. Okay. The functional diversity means through carbon profiling, biolog based. Fatty acid methyl etherate, phospholipid fatty esterase, microbial biomass, carbon, nitrogen, soil and microbial enzymes. Like this we can assess in culture dependent. Culture independent means we cannot culture. See, only 1% of the bacteria is cultivable, 99% not cultivable, means no media is there so far. So how can we assess the uncultivable bacteria? So one technique is there that is called temperature gradient gel electrophoresis. Means directly from soil or from root we isolate the DNA, and then in the second slide you just remember there means we run. Okay, so there means different patterns they are giving, based on that pattern we can know. Otherwise, and after that pattern we cut and sequence the 16S rDNA, and then we come to know bacteria, which kinds of bacteria it is there. Otherwise, target based, if gene is some there, we know, like NIF gene. Okay, the NIF gene, we target NIF gene on whole genomic DNA. Okay, and then we come to know how many types of diazotroph or nitrogen fixing bacteria are there. Okay, otherwise whole metagenome sequence we can go for. Okay, sequencing, metagenome sequence is there, but it is too expensive. Then coming to conclusion. Okay, overall summary is, means endosymbionts act as a biofertilizer and biocontrol agents for non-legumes. I discussed long-term effect of organic fertilizer in combination of inorganic fertilizer formally balance the soil microbial community. Elevated temperature, elevated carbon dioxide, salt alter the soil microbial process. And the last one, beneficial extreme tolerant microbe also play a pivotal role to alleviate a biotic stress in rice. And I am really thankful to my all MSc and PhD students, which is written here. Thanks a lot for your kind attention.
Okay, thank you very much. Thank you very much, sir. Naidu sir and Upendra Kumar sir for chairing the session. Now I would like to request our VC sir to honor our chairman and co-chairman of this session. Please, sir. Thank you, sir. So technical session 1 is over. Okay, thank you very much.